have you take away the messages from today, because it's not a data type that you've seen a lot before, many of you, but the value proposition is there. And that's my other mission today, is to make that part clear to you in the few minutes we have. And so I'll use the language of chromatin biology to do about half of that job, and I'll sprinkle in some recent developments. And there's a central technology development I also want to make you aware about. And so the structure of the talk has 4 points. Proteoforms define that word. It's not just a word, it's a movement. It's a group of people, many of them in Europe, focused on getting really precise about protein molecules, the composition of protein molecules, and doing that with complete molecular specificity. And then taking that to the next level, which is this notion of top-down mass spectrometry. It's a philosophy of how to analyze proteins and proteoforms, and taking it to not just whole histones and sequencing them and their combinations and modifications, but at the nucleoform level, or how they co-occur in, of course, the octamer that underwires so much of life. I'll give you a little sense in a couple slides about AAV analysis and what's possible to do, and as we continue to develop automated pipelines, scalable pipelines for doing what I'm going to show you today. And if I'm at all successful, I'll hit you with 1 or 2 slides about a large-scale big science project akin to sequencing the human genome, and that is a provocative, big idea, sequencing a reference set of proteoforms. But that is, and I'll steal my own thunder here, that's how many of us believe that we can get an AI training set to do what AlphaFill and other kinds of concepts to really get a training set of functional PTMs on the right isoforms and do that well. So let's begin. This proteoform concept came out of a meeting in 2012, in not far away, in Portugal. A group of about 50 scientists, supported by 4 vendors, got together and said, well, what are we talking about? Isoforms, protein variants, how do SNPs, coding polymorphisms fit in there? And then, of course, PTMs, how do they all combine? And that now is in a single word. It's reasonably well adopted. We're starting to see its penetrance in different fields. And I really hope structural biology can be that partner in a unified voice in science. Always hard to achieve. But from a single, say, human gene, one of the 20,000 or so, you, of course, have different isoforms. And then the combinatorial possible modifications that all co-occur on the same protein molecule. Geneticists, of course, understand this very well with the concept of haplotyping, and that's, of course, enabled by long-read genomics. So let's get long-read proteomics up and going. And there's a worldview problem here, that when you see PTMs and you see so many in mass spectrometry-based proteomics, they're so proud of having shown you our field created tau with 65 phosphorylations on 6 isoforms, and you're like, okay, so which ones are functional? Those who do proteoform analysis have a different worldview, by and large, that all those 65 phosphorylations can't co-occur. That's just too many ATPs burning, and evolution would not have created that inefficiencies. So when you think about high stoichiometry co-occurring phosphorylations, those who do this kind of analysis see a much more tractable landscape, and therefore a straight line to a big science project. So here I'll give you a framework how to think about this. Bottom-up proteomics, that's of course the domain of trypsin digestion largely, or other proteases. And yes, it has become quite general, quite well maturing now into the 2020s, all the way from its origins in the 90s, right? That's 30 years running a 4 to 5 billion euro a year market to develop that technology in all of its incarnations, including like bio-ID, turbo-ID, proximity labeling. That's just one offshoot. Cross-linking we saw a little bit. All of those technologies are amazing, and wouldn't have had it any other way. Great for discovery of PPIs or creating data sets that are scalable, useful, understanding the logic of how cells function. So, but for many years, myself and others have been saying, well, for reasons that I just articulated to you, we could go to the next level and have a new unit of measurement, not the tryptic peptide, but the entire proteoform as the unit of measurement. And then the same kinds of arguments are made when you deal with whole complexes. And that the key thought here is in the lower right, that if you strain to go to these higher levels, you'll be rewarded. You'll be rewarded by the functional detection of functional isoforms and PTMs, proteoforms that drive the underlying biology, and you'll be able to prioritize your work moving forward. The world, by and large, is not operating in a proteoform-informed way, and that's what we seek to change. So here I'll tell you a little vignette about a new technology that makes it significantly easier to do proteoform discovery and dynamics, and that is this transition to single particle mass spectrometry. And it's useful in both denatured and native modes using electrospray. I'll come back to define those terms if you're not familiar, but I bet most in the room are familiar with that. And it involves a shift in that mass spectrometers, they never really lived up to their name. Mass spectrometers. No, they were M/Z spectrometers. That's what they measure, either by frequencies or by various other ion physics. But now we've actually been able to live up to the name and produce, no matter what the sample is, with very few limitations, you get a mass spectrum in the mass domain. And what does that do for whole protein mass spectrometry? Well, it takes, for example, a 4-protein mixture here with overlapping charge states. So when you electrospray a protein into the gas phase, it of course adopts many protonation sites. pH 3, typically you're going to protonate lysines, arginines, and histidines, and you have 4 proteins, and they all have overlapping charge states. You can deal with that, okay? Sure, you can, and you can deconvolute, generate masses of those 4 proteins. But as soon as you start to get 40 proteins or 400 proteoforms in a single mixture, you can no longer do that. Or to go to very large mass complexes, like 2-4 megadalton complexes. So this is the innovation, is that now the mass spectrum is produced. I'll tell you a little bit about how it works, but in the Q&A, maybe more if people are interested in it. But here, it's a simple idea that, okay, 4 proteins, 4 signals, and the mass, there isn't a roll-off in the detection at high mass. So this technology gets better the higher mass it is, and it's because of how it works. So here's a native tetramer sprayed with native electrospray, and you see the different charge states, so 50+ charge state. And this is obtained classically with ensembles of ions. You would get enough ions, 10,000 ions, in an Orbitrap. This is all with Orbitrap, Fourier transform mass spectrometry. And the way that you would generate signal is the constructive interference of these different frequencies, spinning around in a motion I haven't shown you, but I will. And their frequencies are different. So these things are spinning at lower frequencies, higher frequencies to lower mass to charge. The frequency precision is incredible. It's 1 part in 10 to the 9, okay, 1 part in 10 to the 8, at least. And so those frequencies are just incredibly precise. But we've changed the nature of the readout. We've changed the nature of detection of signal. So the first thing we had to do is dilute the sample by about 500 to 1,000-fold. That has major implications, and it's one of the advantages over here. So when you do that, it was known that in Orbitraps, you could detect a single particle. You could detect a single ion. The sensitivity wasn't the problem. It was defining those pesky charge states. So you can see, actually, what these are. Now, if these are 50 ions, we let 50 ions into the trap, okay? And you can see where there's abundant charge states. You see a few isotopic peaks. Here's a single isotopic peak from that homotetramer, okay? And so in that single peak, for example, what we did is by 2018, we knew this could work. This was joint with Thermo Fisher Scientific, co-invented technology, and it was just released recently. But what we did is just say, okay, we're going to integrate, we're going to create what's now called a story plot, and we're going to integrate the signal over time. So this could be 0.5 second to 2 second detection period, and that slope is proportional to the charge state. So that's a 48 plus ion. There we go. A 49 plus would have a slightly higher slope. A 47 plus, slightly lower slope. They're very stable. They can be calibrated and stable over months, periods of time. So now the basis of signal are a single ion itself. You no longer need constructive interference. And that has a huge effect. So here's 4 ions undergoing the process. Here's 4 ions, different slopes, and spinning at 4 different frequencies. Extrapolate the slope, generate your now mass spectra because we know the charge on all those ions. So mass to charge is frequency. Right, so that's as much as I'll tell you about the method, but it just has major ramifications. It's drastically accelerated what my lab's able to do, both because of the low abundance, required low amount of input sample. So we've combined this. I have a financial conflict with this startup company, but I'm excited about it because we've combined it with sample prep. So we're automating sample prep, sticking it next to that technology I just showed you, which is called direct mass technology by Thermo. And so this is basically a protein washing machine. It's a protein concentration device and buffer exchange all in the same format. And so you get these focused bands. This is a process called chromatofocusing. So you can basically take low nanograms amount of protein or protein complex and get it ready for the mass spec. And so it focuses here, 20 to 100-fold sample concentration, and then you can mobilize back up through this molecular weight cutoff membrane. It's basically a fancy version of those spin filters, but it's industrialized process. And so we've been using this a lot for integrated structural biology. Just a few papers here, or for immunoprecipitation, automating across hundreds of patients for ApoA1 proteoforms, the nanodisk type protein. There's 18 proteoforms of that in that paper. And then this is from SARS-CoV-2. We have a new kind of serology where we use the RBD, part of the spike protein from SARS-CoV-2, do what everybody else does, which is to take a 50 microliter plasma sample and let the antibodies, the IgGs mostly, bind to that bait of the spike protein, and then look at their clones that come out that bind. So IgGs are then reduced to light and heavy chain, and we get these new patterns. We published 4 papers on this, and so it's serology, so you can get the amount of the IgGs, but because of the way that this new mass spec works, you can get single clone resolution. You can get the individual IgGs, and there's distinct proteoforms that bind. That's a new level of specificity, and we don't quite know what to do with it in the world of serology yet, but it is a technological boost. Well, what most of you are interested in, I think, is the native mode of analysis. And so let me get to that, focused on chromatin, and an area, of course, that's rife with a lot of activity looking at complexes bound to those nucleosomes. But as I mentioned, the talk's really about the decorations on proteins and how to find them more efficiently and the functional PTMs for people after. So chromatin is a great space where that has been decades been theorized since 2000, that combinations of these modifications co-occur to really drive all the central processes of gaining access to the genome, transcribing it, replicating it, etc. So yeah, let's apply then what I've mentioned to you. Proteoforms, how many of them are there on these different histones? This has been largely mapped out now. And for histone H4, for example, where are we? Here we are. Histone H4 has lots of different modification sites. Minimally, you could say, well, how many proteoforms are there, right? So I have all these different sites known. There's one SNP in the human genome. There's 16 different genes of H4. They're all identical sequences. The answer is that if you do the math, you get to about 50,000 possible proteoforms of a histone H4 in human beings. How many does biology populate? Answer, within the first 4 logs of dynamic range, 75. Now that doesn't incur all the low abundance loci and other things. It doesn't combine all of that. But the dominant canonical set of human proteoforms, at least in cancer cells, many different cell lines, some primary samples, a shockingly low number, right? That's the worldview I mentioned earlier, that people think, okay, they're used to looking at histone H4 with all these, and what are we proud about? We're proud about proteinylation and isobutyrylation and, things that are of quite low abundance. And yes, if you go deeper in the proteomic iceberg, you can get down to about 180 or so proteoforms. And, going down with bottom up, you can dig out things that are biochemically real, and then the question is, what are their functions? What are their loci? And that's at least an entree into this field of using denatured top-down for histone H4. For histone H3, of course, it's much more complicated. You have 3.1, you have different isoforms, 3.2, 3.3, and there you get into low thousands of proteoforms, up to tens of thousands for the highly modified states, which carry up to 18 co-occurring methylations at their most complicated. Right, and so this has been a small but active field, and using all the basic technologies, and then looking at these proteoform patterns after drug development, knockdown. Of course, there's many drugs that target histone modifications. Let me give you our data type for histone H4 from human HeLa S3 cells. So there's the profile of the H4 proteoforms. Half of all nucleosomes contain one that has an N-terminal acetylation and a K20 dimethyl. That's present on half of all nucleosomes. And then you have a smattering of others. We've done many papers on this, like the progressive mono, this is 14 Dalton increments. So the progressive monomethylations that occur on K20 throughout the cell cycle, that's been mapped out. This one now has an internal acetylation on 60% of it is plus 42 Daltons, and 60% of it is K16 acetyl, and 40% is K12 acetyl. And so those are actually two proteoforms under that mass peak. So this is all like 50 papers worth of studies, and of course if you inhibit deacetylases, you get a very different pattern. You get a hyperacetylated state here. And so other ones, this is yeast, which has a minimal form of apoptosis. If you treat it poorly and put it in hydrogen peroxide for a couple hours, you get this condensation of chromatin, massive effect of heterochromatin formation. This is what it looks like through our lens of this massive deacetylation. These are all 42 Dalton differences, and you get this massive deacetylation with this phenotype. So it's a bird's eye perspective, and throughout evolution, at least in Saccharomyces cerevisiae, you have an incredible, this result is really cool from years ago, but how did the dance of acetylases and deacetylases that maintain this profile, they get it perfect. They get it perfect in that you have a C to N zipper effect. That first acetylation is all right there. And that next one, it's all K12. That next one, all K8. Next one, all K5. It's maintained perfectly in these yeast. I mean, that's just a result that I found striking, and throughout evolution, there must have been a lot of selection for them. So moving right along, let's do this now at the whole intact level. And again, I'm determined that you take away something from this lecture which has precious few cryo-EM or X-ray, or no tape drive with X-ray microcrystals. But we do have, this native MS, which is a big shift into this domain where even a single protein, right, you end up with fewer of these charge states. So it ends up getting easier, in some sense, to get data. And this is why it works with integrative structural biology, and why I would promote you all to use native mass spectrometry to just see what compositions you have, either before or after you're getting your cryo-EM data. Because you can do this now as well. So once you get the intact complex to fly, so like here's an intact nucleosome, you can controllably disassemble it. you can eject out subunits and then sequence the proteoforms that are your subunits. That's the takeaway that I'd like you to pull away. And so let's just do this a couple times with different nucleosomes. Here's a recombinant nucleosome with your basic 147 bases of DNA wrapped around. The intact complex is about 200 kilodaltons. And then on the right are these ejected subunits. So what we did is we just tickle, if you will, or bash apart the complex with collisions in the gas phase, and out pop the subunits. In this case, the histones, the 4 core histones. And then once we have those signals, we can sequence those proteoforms. And the nature of our data are these blue flags, these fragment ions, which you could kind of think of as tryptic peptides. There are sequencing equivalent, right? But we use no digestion throughout this process. There is no protease involved in this. And so here's, again, a picture of how this works. Tetramer comes in, you measure the tetramer, and what happens is by some beautiful fluke of nature, and we actually got a big proposal on this in 2011, that when you fragment these gas phase cations, and they're in a complex, non-covalently held, when a subunit escapes from that complex, it steals a bunch of charge. This is known as asymmetric charge partition, and you can see what happened. I was at M over Z 6,000. Now I'm at M over Z 2,000. So it stole a bunch of protons, which is great if you're going to then take the subunit and further bash it apart. So here's my subunit, and now I'm going to fragment it into these terminal pieces in top-down mass spectrometry. So there's the fingerprint. There's the data that we use to partially sequence proteoforms. Those are the blue flags. Those fingerprint pieces, each little line on the fingerprint is one of these blue flags. And I could go into more in the Q&A, but that's probably enough for how this works. Let's go ahead and use it. Here we're just in vitro treating with, of course, an acetyltransferase or a methyltransferase. And I could take you through, but I think given how much time I have left, let me keep going. But we can do what we said we'd do. Sorry, this laser pointer's a little weak, but yeah, there's an acetyl. We can localize it. If you know anything about PCAF, you know K14 acetyl is where it should write its mark. And PRC, it should write its methyl mark 1, 2, and 3. EZH2, the subunit of PRC2, can write all 3 marks at K27 monoditrimethhyl. And so now we did this on endogenous nucleosomes. We're immunoprecipitating out an engineered version of H3.3, an isoform that's known to be in permissive areas of transcription. So, and then we create mononucleosomes, we immunoprecipitate, and then do what I'm showing you is what we call nuke-MS, or nucleosome mass spectrometry. And when you do that, the H3.3 in more permissive regions have a 6-fold elevation of H2A.Z, a specific one of 15 different H2A variants. And that's the evidence for that that's now published. Here's it is corroborated with ChIP-seq on 2 different antibodies, co-occurring with our flag-tagged H3. So, where's this going? Let me just give you a sense of something that's maybe more interesting to you, which is whole AAVs or whole complexes, and looking at their cargo. This is something that's, of course, now FDA approved. It was first approved in Europe. Most of you probably know that. And it's a ramping area for drug delivery because of this pathway on the right that I'm skipping right over. Of course, preparing AAVs is non-trivial, and then making sure that you characterize your biotherapeutic, also non-trivial. So there's many steps involved in the processing of AAVs, and this is more of an engineering type project. What you do now, if you want to use native mass spectrometry on an AAV, you take your sample, maybe there's 10 to the 10 or 10 to the 11 particles in there, and you spray it, you electro-sneeze it into the gas phase, and you're running with these little manual needles. So this is the nanospray, basically 10 micron little hole, electro-sprayed manually. It's kind of very detail-oriented work, it's all manual, and there's some state of data analysis tools. So we partnered with Thermo to go ahead and automate this with the technology I showed you before, the protein washing machine, be able to take in a sample of an AAV8 or AAV5, what have you, and do the pipeline that I showed you today. And so I don't need to re-explain that, but we're pushing this in terms of automation, but also data analysis, which we have an automated workflow now that works in about 20 minutes. And so here, pretty tight error bars here for empty filled and then the double filled virus. And this is all occurring at about 4 megadalton, I should say. So here's the patterns. These are quite wide. These are 200 kilodalton wide peaks. And the nature of that is some solvent and maybe some molecular heterogeneity for the AAV itself. We need more research to figure that out, but we can get these empty-full ratios and do it in a scalable way on very few numbers of capsids. So this is now approaching below 10 to the 9 capsids per input sample, and that we think we can improve by another 5-fold. So that just gives you a sense. This is unpublished, but soon to be published and presented at an upcoming conference in a few weeks. So let me just end, about 3 minutes left. Let me just end with some perspective about this. I know that those data types may be foreign to many of you. The value proposition may not be clear, but I've tried, and we have other papers that try to address the function question, right? If I have a short isoform and a long isoform, and I have different modifications, phosphorylation on one or acetylation on a long isoform, I really need to know that. Compositional proteomics can really help, in both for structural proteomics, but also single-cell proteomics. And this is where our lab's going with this in one direction, but in another is just to be able to sequence the human proteome. Rolls right off the lips, right? Can we generate a reference set of what is an estimated about 100 million proteoforms from the 20,000 human genes? Now, I understand that that's a provocative thing, that big science project makes labs that are hypothesis-driven nervous. And I get it. But why is this information argued to be foundational or fundamental? Because when you know this, you know the stoichiometry of the mods. You can capture the dynamics of the system, and you have clarity about isoforms, their PTMs, and how they progress through disease. That's the window on biology we don't have. So there's a growing crescendo of people arguing that a human proteoform project would be foundational and fundamental. And then the question is how to proceed with such a sequencing project. We also don't have single molecule protein sequencing, but we do in genomics. And so this disparity continues, and many, of course this is a name, you shouldn't name drop unless you have 20 names to drop. We actually have 60, and I've been trying to whip people more into a frenzy about this, which is what it would take to a big engineering kind of project. But there are a lot of private sector companies who have taken the successes and the actual actors from that success in genomics and moved it over to proteomics. So companies like Quantum-Si or Nautilus, right? These are new companies, single molecule protein sequencing companies that stand ready to partner on this. And so while I could only give you the very beginnings of proteoform landscapes, and classic one I think about is the KRAS. So KRAS, we started off with 11 proteoforms, and now we have 39 proteoforms, and we're assigning function to them slowly. But you can develop technology, deep proteoform sequencing, to really give your perspective about a city like Chicago, or our biology and all the cell types in human beings. And I'm happy to describe that project more. There's tons of literature out there about it. But for now, I'll just yield to questions. Thank my group and sponsors, so Thermo Fisher, IPT is a startup out of the group, and these folks really made it happen. Luis is now at Genentech, who did a lot of that chromatin work. So in the United States, you can imagine who might fund this. This is who it is. NIH, we run a National Proteomics Center for National Institute of General Medical Science, the third largest NIH institute. Thank you for the invitation to Cascais. Have you seen this place? You will find me at the beach or the bar somewhere in the near future. I'm staying through Sunday, so thank you for the invitation, and Carlos, the organizing committee, and I hope you took away something from today's talk. Thank you.